Hi, everyone. Welcome to Foresight's Neurotech Group. I'm super, super excited today to have Andreas Tolias here. He's a neuro AI scientist, and your Stanford Lab's mission is to build a less artificial intelligence, which I love as a tagline. You're not only a Stanford professor, but you also really chair co-direct the Enigma project, which is a nonprofit research organization that is based at Stanford and that is also leveraging deep learning for neuroscientific insight. And so you really are trying in all of your work to combine this kind of systems engineering, computational lens on neuroscience and vice versa, and to really decipher like the principle of intelligence at a pretty fundamental level. So it's really exciting to have you here. I've had the brief pleasure of meeting you in person once, and otherwise I've really enjoyed your core memory podcast with Ashley Vance, and that was really fantastic. So hopefully we can dig into some of that in your talk today. So thank you so much for joining. It's a real pleasure to have you, and please take it away.
Thank you so much. And again, my apologies I just came to visit Houston here and there was a problem with my wife in the, with my, with my Wi-Fi in the house. Do you see everything okay?
Perfect.
And you see it going forward?
Yep.
Perfect. Okay, great. Thank you so much, Alison, for the invitation. I've heard a lot about your foundation and what you guys are doing, and have even seen some of the stuff online. And it's very exciting to talk to this very diverse and very exciting and interesting group. So, in some ways, a big goal in neuroscience is really to understand the relationship between the outside world and how we interact with it, and our ideas about it, our perceptions about this outside world, and what's happening inside our brain. So you can think of it as like there's like the universe out there and how we interact with it and what we do with it, what we perceive with it, and then what's happening inside our brain. So we're looking for like correspondences. And our brain is trying to understand, create this world model. And the better the world model, the better it's able to survive and make predictions. And in some ways, that's what inference and intelligence is. It's about understanding the world and given some constraints and some goals, try and survive and take action in the world. Now, the brain, what is remarkable about the brain is that there's very many levels of analysis. And in some ways, it's a little bit more different than other fields of science, including, let's say, biology or chemistry. Where in neuroscience, you can study the brain at the level of molecules and use all the molecular biology tools. You can look at cell types, connectivity, the circuits, go all the way down up to the activity in the brain, and more like the behavioral level or psychological level. So you have a span of levels of analysis. And that is, in some ways, makes it very exciting, but also very challenging. Unlike other fields where it's more well defined, if you study, let's say, quantum mechanics, or if you go and study organic chemistry, there's certain level that you are mostly focused. Whereas in the brain, if you say, I just want to understand the brain or understand these correspondences between the outside world and what's happening inside my brain, it's a more complex thing. And here is some prior work that we've published where, for example, we looked at the cortical microcircuit. This is one of the first, now it's 10 years old, canonical mapping of the relationship between all these cell types. The brain, you can think of it as a forest that has many different types of trees and plants, and it's not even clear what's the logic of this organization, how many there are, but they seem to be connected in stereotypical ways. It's not just a random connectivity. So at the same time, we have other intelligence systems really in the last few years, I would say more pronounced in the last, let's say, 5 years, which include like large language models, computer vision models, and models that make predictions about things that we humans are not good at, like, for example, protein folding. And these are also very intelligent systems. I'm sure you've all chatted with large language models like ChatGPT, and you can get a sense that these are really, for the first time in the history of humanity, we don't just have access to biological intelligence system, but these artificial systems that we humans engineered. Okay. And they're also trying to do this thing. They also have a neural network. They also have activity, interestingly enough. This is what works now in AI, unlike other ideas that were explored in the 80s, the 90s, and later on. And they're also trying to find correspondences between the outside world and their brains. For example, if you talk to a large language model, it has a sort of an understanding of the structure of language and human psychology and is trying to interact with you, or has some understanding of a scientific field and is trying to give you feedback. There's something very remarkable that didn't have to be that way, that makes this field of neuroscience and AI at a very pivotal time in the history of humanity. And this is, I will call this the principle of universality. And it basically boils down to the following observation that we and others have observed for the last, let's say, 5 years. The first is shown here in purely artificial neural networks, where these are three different neural networks, and they're trained on object recognition tasks, like to recognize cats from dogs. And what I'm going to say now is even true even if you train them to do other vision tasks, like, for example, segment a scene or predict the next frame of a movie. And if you look at them at a certain level of, if you look at these three networks, if you look at their individual neurons at a certain level of their processing, they seem to be doing similar things. What is shown here is stimuli for each neuron, each square. You can think of it as the area in visual space that the neuron is receiving information. And those gradings are the patterns that it likes to activate itself. It's like what this neuron wants to do in life. And you can see that despite that these networks are trained with completely different architectures, they don't have the same connectivity, let's say, they don't have the same, they don't quite have cell types, but some sort of do, maybe one or two cell types. They're just different beasts, let's say, mechanistically, the way they work inside. What they compute, if you look at what a neuron ends up liking to activate itself, is the same, okay? So this is the sort of concept of universality. And what is even more remarkable, if you take a monkey, which has also a high resolution visual system like us and like these neural networks are trained on, despite that it's built from completely now different hardware, it's not built out of silicon, it's built out of carbon, has all these cell types, has all these molecules changing the synapses and so on and so forth. If you look at also what these neurons like, and these are the neurons on the left, they also look very similar to these completely different intelligent systems. So there seems to be some sort of convergent evolution here between biological systems and AI systems. And this is quite remarkable because it just happened that when AI in the last 5 to 10 years started working, we started making these observations that there is this universal. And this level of analysis, what I'm showing you now, it's a specific, you remember how I told you there's many levels of analysis to study the brain? It's a very specific level. It's called the representational algorithmic level. So broadly speaking, David Marr defined three big levels. There is the implementation level, how something, a computation is implemented, and that includes the molecules, the biophysical models, the connectivity, all the cell types, all the biological substrates, the ion channels. It's like the whole complexity and beauty of biology. And then there is like information processing level, which is what I'm showing you, and it's saying that this neuron likes to respond to this Gabor-like patch or these blobs in visual space. And then there's the behavioral level, which is our perception, our intelligent behavior. So in the last, what we've seen is that there is this representation level. Now, what is interesting, this level of analysis has been discovered about 100 years ago by this guy here called Lord Adrian, who was at Cambridge University, that discovered the action potential. About 100 years ago, we just knew there was electricity in the brain. By accident, one day, he discovered that these neurons are encoding information by spikes. They have this like binary code. Okay? And since then, it's been used by many in the last 100 years. And as you see, many Nobel Prizes were awarded for this. And it's a disproportionate number of Nobel Prizes for any specific subfield to understand how the brain works at this representational level. And these are the most recent Nobel Prizes, now 10 years ago, where the Mosers in Norway discovered that when the rats navigate in the world, and John O'Keefe earlier on in the 70s, they formed these like spatial maps called grid cells or place fields. Okay. Now, when we train artificial intelligence systems to do these kind of tasks, people find these levels of representations. Okay. Now, what is the dirty little secret of this whole field? And for those that are neuroscientists, they understand this, but even people that are, like, say, computational neuroscientists, and they're not experimentalists, they may not be aware of it, is that these discoveries didn't happen because someone was very smart and had a very good hypothesis of what to look for. In fact, it's the opposite. People were looking for something else, they designed their experiments, and by accident, they discovered these things. In fact, Lord Adrian, when he discovered the action potential, he did it in an afternoon, and he thought it was an artifact. And it was a bug in his system that he was expecting to get a continuous voltage signal. What he was recording, he was getting these like basically abrupt changes, and he thought, okay, it's some sort of noise in the system. And there's a very nice documentary here. This is Hubel and Wiesel, who got the Nobel Prize in 81. And they describe how they discovered this edge detector, which in fact is the inspiration for the Fukushima network, which then led to the convolutional neural networks and the AI revolution. So if you take the AI revolution that happened with the AlexNet, that I'm sure many of you have heard about it, you can trace its inspiration to this experiment here that I'll show you. I'll play a movie now that Hubel and Wiesel did, and they discovered that it was accidental discovery. And this was based on the following thing. They were using spots of light to figure out what these neurons in the cortex are activated. By accident, one day, the projector they were using created this thin, faint line because they didn't bother to clean it and it was dirty. So there was the background. This is in the days where there was not even monitors, okay? And then they noticed that this is what was activating the neuron, and then they discovered the edge detector, and then they understood this concept of receptive field organization, and then led Fukushima and others come up, and Yad LeCun with convolutional neural networks, and then, I don't know, a few decades later, when GPUs became available and you could get large-scale data, and we had ImageNet, we basically built AI. This is the truth. So it was an accidental discovery. The reason why this is a very complicated thing is that if you have a monitor that has 256 by 256 pixels, and each one you turn on and off, I'm sure you remember from math that you can generate 2 to the 65,000 possible patterns. There's only 2 to the 300 electrons in the entire universe. So this is an infinitely large number. And even if you're restricted on the natural image manifold, it's very hard to characterize in an experiment what these neurons are doing. Now, if you take into account behavior, multimodal integration, cognitive states, it becomes a daunting task. So this shows you here the macaque visual system, and this is a simplification. It has in the order of 20 to 30 visual areas, how you count it. About half of the macaque brain does vision, and about 30% of the human brain. So it's a very complex system. And each one has different cell types that may be doing different function. We have different cortical layers. And if you look what we know in the last 100 years, it's very sparse. It's basically what I showed you on that line of the Nobel Prizes. That's pretty much the fundamental, let's say, principles of organization. This is a very simplified version of what I'm saying, but just capturing like the big picture here for a more general introduction. So what I will describe you now is what we've been working on in the last few years to get around this problem and create for the first time in neuroscience, in the study of intelligence in general, a systematic way to decipher the algorithms that give rise to intelligence. And this is really based on two parallel revolutions happening in the last 10 years. One is the neurotechnology revolution, and a lot of it thanks to the BRAIN initiative that was funded by NIH and other organizations, and that's how we got the money for the Microns project that I'll discuss towards the end. We have now technologies that for the first time we can do very large-scale experiments. For example, this is a microscope that we recently built, collaborating with Ali Pasha Vaziri, that developed it at Rockefeller, called the light beam microscope, that can record up to 1 million neurons simultaneously. Here's the Neuropixel technology from IMEC in Belgium, which is the most advanced semiconductor research foundry in the world, that build these neuropixels that we can implant in monkeys, and even humans could get high-resolution electrophysiological data. So we can get large-scale data at orders of magnitude that 10 years ago was impossible. Just to give you a number, 10 years ago, the best labs in the world could record maybe 400 neurons. Now, with imaging, we can go to 1 million neurons. With neuropixels, we can go to tens or maybe soon hundreds of thousands of neurons. And before, we could get maybe 15 neurons, well isolated. So we've got an exponential increase. But despite this, we have not gained any deeper understanding. So think about it. For the last 10 years, we went from 100 neurons to 1 million neurons. But if you look at like our fundamental principles of understanding the brain, they remained stagnant, what it was 2 decades ago. And the reason is because we did not have the tools to draw knowledge and principles from data. But then at the same time, what was developed was AI, which you can think of it as a very powerful statistical tool that excels at ingesting large amounts of data and building predictive models. And this is what we put together in my lab to develop this paradigm that we call Inception loop, inspired by the movie, and you'll understand why. And it's based on this idea of building digital twins of the brain, or brain simulations, at this representational algorithmic level, where we have large-scale data and where we can use the AI tools immediately to build these models, and not rely on developing new tools that would maybe take another decade to develop, like going down to physical models of the brain and so on. And I can discuss that towards the end. So Inception loops have 4 steps, and this is like one thing to remember from this talk. The first step is you do a large-scale data collection on the functional level. Here, we're not going to worry about the lower level, the molecular or anatomical level, the connectome, cell types. We're just going to record as much data as we can functionally, ideally at single cell, single spike resolution, because that's the code of the brain. Now here, these are and we're doing it in different ways. These are calcium imaging data collected with a 2-photon microscope or a 3-photon microscope. But we basically place a mouse here, and I'll show you also data from monkeys, in front of a monitor, and it's just watching passively in this case, or we can train them in tasks, a large amount of images. So once you collect this large-scale data, the step number 2 is you build a functional digital twin. So what you want to do is, instead of predicting the next word or the next token, or predicting if it's a cat or a dog, which is what AI is doing, that's the whole field of AI, is basically taking human behavior and copying it on a chip. For example, you just take language and you say, what's the next word? We humans spend 4,000 years creating this data, or people took out a camera and took a video and you say, what's the next frame? So you just take human behavioral data and you copy it. Here we're doing it at a different level. We're doing it at the neural level, which is what's happening inside the brain. So here we're building, again, a digital twin of the brain, the way that AI is building, if you think, a digital twin of intelligence, of human intelligence, and maybe we'll surpass it. But here the input, the input is again video or images, but we're predicting the neural activity that the brain generates. But the way it's doing it is going to be very different. It's not going to have the same cell types, it's going to have the same connectome. And because these neural networks are universal function approximators, and you have a lot of data, it seems to be working, you can reproduce the function in a different way, in a different substrate. And we call this substrate independent intelligence, or the way this thing is computed. Okay. Now, the beauty of this is that this is the second step. You collect a lot of data, you build a model. Now, once you have this model, you can do an experiment. It's not that it would have been impossible to do in the brain, almost. One thing to think about it, instead of, if a graduate student spends 5 years doing experiments and each day spends 8 hours, has a limited amount of experiments it can do, or because of our own lifetime or the animal's lifetime, we don't live 100 million years to do experiments. But if you have a faithful digital twin of the brain, you can parallelize it, you have enough money in a GPU, and you could run what it would have taken 100,000 years in a few hours. So you can extend the experimentation at an unfathomably high level that 5 years ago would have been not even a dream. On top of it, because these models are differentiable, and this gets a little bit technical, but for those who are working in this field, you understand what I'm saying, you can also do optimization in the model in ways that would be very hard to do in the brain. So here's the first experiment we did. We took this model, we took one of these neurons, and we did the Hubel and Wiesel experiment, but with no hypothesis. And we asked the question, what is the optimal stimulus that this neuron likes? And when we did this, we replicated the serendipitous discovery of Hubel and Wiesel and found that these specific neurons like some sort of grading or grading patch in a specific part of his visual field. Okay? But what was interesting is when we did this, we found that unlike what Hubel and Wiesel predicted, at least for the mouse, many neurons validated this principle. We found many types of neurons liking a very diverse set of features that look like patches in natural images, like some like corners, some like checkerboards, and so on and so forth. Because now the fourth step is not enough to do this discovery in the model, because the model can hallucinate and can fool you. But because in my lab and also in the Enigma lab, what is very unique about what we have at Stanford right now is that we have the entire pipeline built from people collecting the data to building the models, doing the mechanistic interpretability and the analysis, and we can close the loop, and that's why it's called Inception loop, from the Latin word incepire, which means to synthesize, because the model is like synthesizing the stimuli or synthesizing ideas. We can now test them back in the brain. So we can do this while experiment is being run almost in real time, or we can come back the next day, bring the animal back under the microscope, these are wake behaving animals, find those neurons and test them and see, are they doing what the model told us they are doing? So this closed loop experimentation, where AI is at the center now in the large data collection, it opens up a lot of possibilities. So I'll just go through a few discoveries that we made. First of all, in the monkey is different than the mouse. Here in the monkey, that Hubel and Wiesel were pretty much right. Here is another thing where you see this principle of universality. For example, Chris Olaf from Anthropic discovered a couple of years ago that these artificial neural networks, they have these low high frequency detectors that we think they're important for segmenting a scene. And we find something similar even in the mouse. And if you take this now into a monkey, and these are monkey optimized images, that they're way more beautiful, right? That the complexity, they're starting to like things that are almost humanly interpretable at the level of these individual neurons. For example, these neurons, and this is work done by Nico, Sophia, and Katrin, shows that, for example, they like these eye-like patterns and so on, or like fur. And they seem to also be clustered into different groups. So it has to be, it's almost like the periodic table. There is like some logic. We still don't know what that logic is. But now for the first time, instead of, if you imagine Hubel and Wiesel having to spend time to accidentally discover these features, that's impossible. So now for the first time, we have the technology and we have the method to systematically decipher the entire visual system. And now we want to take it into motor control and also more cognition. And we can apply these methods in humans too. Okay, so I'll just show you one discovery that was made completely by this method. This was a paper published in Nature a couple of years ago by Catherine Cadwell, which was asking the question, what happens to your brain when you change your internal cognitive state, okay? And what I will describe to you now is in mice. We're doing this now in primates that are more closer to humans. But for example, you'll know, maybe some of you now are really paying attention to what I'm saying, or some of you are thinking about what they're going to eat for lunch. So your brain, how is it changing the way it's processing information? We don't know. Are these neurons are the neurons that were supposed to firing to edges? For those who are thinking about food, do they look at an edge and they think maybe it's I don't know, a banana or something, if you're going to eat a banana. We don't know how the brain rewires itself when this thing happens. So what Catherine did, these were the first experiments, I said it was published in Nature a couple of years ago, was mice, unlike us, we have a red, green, and a blue cone. They have a green and a UV cone. They're very sensitive to UV light, like deep blue, basically, something that we cannot see. So she built a monitor where, in our lab, where she showed colored images from the mice, built this digital twin, and then in the digital twin, did this optimization under different brain states. And one way to measure the brain states, both in mice and humans, is by looking at the pupil dilation. So now if I had enough high resolution and I looked at your pupil, if it's dilating under the same luminance level, that means you're paying more attention to what I'm saying. And in fact, poker players and others in games, one of the reasons they hide their eyes is for a number of reasons, but it includes also people are very sensitive to watching each other's pupil, and when it dilates, they're very sensitive to their internal state. So she asked this question in mice, and what she found, it just shows you, like now in the model, you see the mouse's eye dilates, and the neurons get tuned to more blue or UV light. So basically, when the mice pays attention, it doesn't change the spatial features that it likes, but it shifts towards blue. And the reason that we think this is important is during mice, of course, they worry about predators from the sky. I'm sure movies where some bird or a hawk or something will come and grab a mouse. They come from the sky. There's a lot of, and they are out during dusk and dawn, the blue hour. And this is a beautiful sunset in Santorini. And for the wind, everybody's like standing there watching the sunset. But when the sun goes down below the horizon, you get this blue light. It's called, due to Rayleigh scattering, you get this UV light. And what my collaborators in Tübingen did, they flew drones during this time and measured the sensitivity of, let's say, a flying bird or a predator in the upper visual field of a mouse, and found that there's more contrast in the UV. And then with some experiments we did, we showed that during this pupil dilation, when they're paying attention, they're much better at detecting flying objects that would mimic a bird taking them. So what I've described so far were digital twins built individually for each animal. What we are done since then, and we just recently had, as part of this Microns Nature package, another paper we call a foundation model. We built a foundation model. This is one of the first foundation models of the brain, where it's trained as a Rosetta Stone to relate, in this case, video and behavior to responses, not just in a specific mouse, but across all mice. This is trained on massive amounts of data, that we've been collecting, and now we're training even the bigger one that's going to be trained on 300 animals and close to 3 to 4 million neurons. But this is one of the first ones. And now it's processing video. And the reason that we call this a foundation model is inspired by the terminology that's used in large language models that originated by Percy and others at Stanford, which is the idea that once you train this model, it's not just good at the task you've trained it on, but it can perform a lot of new tasks. And that, of course, opens up the Inception loop capabilities much further. And I will show you some capabilities. For example, once you train this foundation model on let's say, a cohort of animals, let's say 10 animals, now you bring in a new animal, and very fast you can build a digital twin of this animal. So here it shows you that what would have taken an hour and a half, this is the gray line where each animal is built its own digital twin. If we have this foundation model and we collect 16 minutes of data only, we can build a digital twin as accurate as if we were building it from the animal from scratch. Of course, practically for us, doing experiments opens up a lot of possibilities, because now we can run real-time optimization of the experiment almost within a couple of hours. The other thing is that this model was only trained on natural movies, but then you can show different types of stimuli, even parametric stimuli, random dots, so you can explore the space of cognition, perception here at a much higher level. You can also use this model for brain-machine interfaces. You can decode. The mouse has low visual system, but this is, the state of the art in pixel level reconstruction. This is just raw data recorded from the brain. There's no priors, and you're just reconstructing the video. And we're doing this in monkeys now. And now to finish the talk, I want to switch gears because until now, I only talked about this representational algorithmic level. And I think that is the level that is in the next 5 to 10 years is going to have a direct interactions and possible impact with AI because of this principle of universality. Because at this level now, as I've shown you, we can basically copy intelligence, not just using behavior, but using neural activity on a chip on a GPU at the same substrate as what, let's say, Google, OpenAI are doing, and we can compare these models directly. They are built on the same substrate now, but one is mimicking the brain functionally, but not the way it's doing it. It doesn't have molecules, it's built out of silicon, not carbon, it doesn't have connectivity, it doesn't have cell types. And I think this, in terms of AI and interaction with AI and understanding the brain algorithmically and building digital twins, what this functional digital twin of the brain, not implementation level, not a molecular digital, not about physical digital twin, but a functional digital twin is the most direct path forward to relate this to AI and understand the brain at the algorithmic level. And I can discuss it in the end. The discussion is also maybe related to building also maybe safer AI. However, we as neuroscientists, so basically what I've done here is what I did when I was a graduate student, maybe the same, some or undergraduate actually, maybe some of you discovered the same thing. When you're a student, you don't have money or resources to buy every tool. So if you want to make a toast, you can use an iron board and you can use your iron to make a toast. So functionally, I copied, and I'm sure a lot of you have done the same, the toaster into a different way. It's built from different components, it even works differently, but the product is the same. So I call this a functional model of the toaster. However, we neuroscientists are interested to also fix the brain and treat diseases. So if the toaster breaks up and the only thing I know is functional iron toaster, I won't be able to fix it. And to fix it, you have to go at the level where we have a lot of tools to manipulate the brain. And this is where you have molecular biology, pharmacology, and so on. And this is what the Microns project is about. The Microns project is the largest connectomics data, but it's not just the largest connectomics data in the brain, it also has function of the same neurons. So these data were collected in such a way that has both structure and function. This was a heroic effort that took 7 years to complete, and we published 10 papers back to back in Nature in the last issue, in the April issue of Nature. Okay, and I will show you some of the data are publicly available. It's been publicly available even before we publish any papers ourselves, but it's very rich. The data has cell types, connectivity, down to organelles, through electron microscope. So I will show you one of the main findings in this paper, and I will finish there. As I said, this data are very unique, not because it's very large connectomics data, but because they have function too. So what we did here, and this was Jokun, Paul, Estelios, they aligned the data between the function and the structure. And then we could first ask a very simple question. Could this foundation model that Eric Wang built predict not just activity, but the morphological, the anatomical cell type of these neurons? And this was the work, this is another paper that was led by the Allen Institute, where we've characterized the anatomical characterization of the neuron. This was mostly Nuno da Costa and Casey. And then this model, in Eric's model, as you see here, could predict the anatomical information, despite the fact that this model never saw anatomy. So that's why it's a foundational model, because it wasn't just predicting activity, it could also predict other things. Then what we did in my lab, we took this data and we asked a very simple question. When neurons make connections, what is the relationship between the two neurons? How are they related functionally? To give you the summary here, there's something called the like-to rule that we knew for many years. And basically it says that when an axon, this is the output of a neuron, goes through, let's say, a forest, and there's all these trees that are the dendrites, it will connect to neurons that has the same preference. But what is that preference? And what we found is that if you break down the preference, if you take this model, it decomposes what neurons like into the part of the space that they like something. As I've shown you, these neurons have localized receptive fields. And the type of features, for example, they like red or UV, or they like a vertical bar or horizontal bar, or they like this corner versus that corner. And what we find is that neurons are promiscuous, more promiscuous instead of where they like the features spatially, but they're very faithful to find neurons that have the same features they like. If they like a red right 90-degree corner, they'll try and find other neurons that like that same corner. So this was the rule that was deciphered through this combined structure and functional data and using this foundation model. Okay. Now to finish, to summarize the big one message from this talk, if there's only one message, is that our goal is to find the relationship between what's happening inside the brain, what's happening outside. That's basically the definition of intelligence. So what I've proposed is that to do that, if we build a digital twin, a functional digital twin of the brain, not only we can relate to AI because we brought down the brain intelligence into the same in silico substrate without having to worry about this grandiose detail that we don't even know how to collect at scale. And even if we did, it's not clear. And I'm one of the people that have collected connectomics data and cell type data and transcriptomics data, how to use it, because the best models we're building just built on this functional level, is that it's a way to basically make these links between the world out there, a digital twin of the brain, or an AI system, which is also a digital twin, but is built more at the functional level, whereas these digital twins that we're building are built at the functional and the activity level, and understanding how the brain works. And this could create what we think the big hypothesis here is that the brain, for example, in vision, has intuitive physics. It has an understanding about the world. It's not just competent about the world. And here is a very nice demonstration. This is Sora from OpenAI that shows that despite these beautiful movies that you see generated by Sora, by Evo 3, and so forth, when you make them do some simple things that we humans is trivial or animals have to do with intuitive physics, like for example, the legs cannot move in this weird way because of the joints, these models fail. And that's where we think the brain has this more new type of ingredient that gives it understanding versus just being competent in the world. And I would like to thank our funding agencies that have funded our work over the years, and also the many collaborators and students that have been working on this. Thank you.
Thank you so much. That was totally fascinating. Does anyone in the audience have any questions? Now we'll move into the Q&A. We have about 10 minutes. Micah.
One can imagine building a mouse digital twin based on visual audio inputs like you have shown here, but where it predicts externally visible behavioral outputs. How do you imagine people of the future deciding whether they should use a model like that or a model that instead predicts the individual neurons? Like, which one is good for which task?
Yes. First of all, this model that I've shown here predicts the individual neurons. It just doesn't implement the function the way it happens in the real brain, because it doesn't have the connectivity and the cell types and the biophysical properties. These neurons they have very complex morphology, they have very complex nonlinearities, and so on and so forth. It's like this for 100 years people have been studying that. However, what you're asking is a very cool question, because at the end of the day, there is a bigger question, which is, do we want to worry about the individual neurons, or do we want to study these neural manifolds, like basically the dynamics that are generated? And then there is other technologies like fMRI, ultrasound, EEG that abstract away from the individual neurons and measure brain activity at a different level. I've worked in these other areas like fMRI, and also we're doing experiments with EEG, like high resolution EEG, even intracortical. And my feeling is the following, and my experience building models like that. The brain is working at the level, it's like the quantum level of the brain is the individual neuron, individual spike. And now we have invasive technologies to get this data at scale. So that's where I would put my money right now, and only move to less invasive stuff once I get evidence that I can understand at this other level. Think of it like you're trying to understand human language and you're in a stadium, and you can drop microphones that record hundreds or thousands or tens of thousands of people. But even if you record the conversation of 10 people and you just average it, of course it's going to be correlated when there is a goal or when something happens and when there's maybe a fight breaks in the stadium or whatever. But to get to the language of the brain and understand what they are saying exactly and understand the syntax, you have to go to that level. And my feeling is that there's no free lunch here. We're going to have to go to that level to really understand the code. And that's the level that I'm going for.
Thank you. Are there any? Oh, Allison, go right ahead.
Yeah, I absolutely love.
Congratulations, Allison, by the way.
Thank you. She started screaming right when you started, but I really love the presentation. I have one question. You mentioned towards the end of it that it might have application or implications for building safe AI, especially, for example, the less artificial intelligence part of building artificial intelligence, aka perhaps like not exactly doing it on silicon, et cetera, et cetera, et cetera, in the way that we currently are doing. Could you just go into a little bit more depth?
Yes.
Thank you.
Let me stop sharing and I'll show you guys a figure, but I'll talk about it until I find it. So the idea here is that the brain, as I said, intelligence, a safe intelligence system, one aspect of safety is to have understanding of the world. If it's a savant that just performs well, but doesn't have true understanding, like it makes stupid mistakes, then it's a worrisome, it's an unsafe system. For example, let's say if you give you like a very simple example. If you build, let's say, a game that is playing Go, and you change the size of the board, and it's never seen boards of different sizes, and it catastrophically fails, that means it's not completely trustworthy. Or you have a robot that has never seen the shadows in exactly this way, and it doesn't have an understanding of how shadows are formed in the world, is not safe. The big hypothesis here is that the brain has an understanding. It may not have read every single text, may not have seen every single video on YouTube, but it has an understanding of the world. And this understanding has to do with interpretability, and the model is interpretable. It's basically has a world model that, for example, in vision, has the colors, the shapes, the objects, 3D, the 3D structure, the pose. This is entangled in a beautiful modular way. It has compositionality and all this stuff. And if we build these digital twins that are built out of the same algorithmic principles, then we will be able to build them also to be safer from that point of view. I'm not talking about from the morality point of view, but basically having an understanding of the world and not doing these sort of, let's say, mistakes that you would see in artificial neural networks. And I'm sure you've seen them, like they will catastrophically fail in some way. They have these gaps in knowledge that shows they don't have true understanding. And there's a very nice, I'm trying to find it, the late Daniel Dennett, who is a philosopher, he has this thing before he died on his website that I just want to show you that kind of basically captures what I'm saying here. And I'll just show this and but you can ask the next question if you want while I'm sharing the screen.
Thank you. Michael.
Okay, here is the thing, just to finish this, competence versus comprehension. If you look at this, the thing on the left is a termite mound. It almost looks like La Sagrada Familia. but there's no architects here. These guys are just building stuff that can look very beautiful, but there's no true understanding. For example, they cannot reuse these things and build something that looks like the Parthenon now, like an architect could. So Daniel, who's a philosopher, and of course a bit of exaggerated version, and I think now these artificial neural networks are closer to understanding, but I think it captures this concept between competence and understanding. Okay, we'll go to the next question. Thanks.
Hey,
Hey, Michael.
hey, Andreas, this is an awesome talk. Yeah, I've got a question of why do you need to correlate with behavior? Is just functional recording and neuron morphology good enough to predict cell behavior, predict neuron behavior?
Yeah.
Why do you need behavior from the outside? Why do you need to be recording what the mouse sees, what the organism sees?
Yeah. Okay, so we need to record, let's say in vision, right? If we record what these neurons are responding to, in order to build this model, we need to know what the output is, what the input is to the system. Because, for example, if they're trying to basically have a representation of the outside world, as I showed in the beginning of the talk, so having the characterization of the outside world is very important. Now, the reason we also need behavior, like what they are doing, are they grasping when they see an apple? Or in a human, if you talk to them, you can, with a microphone, record what they're hearing, but then what is the thing they are saying? It's because ultimately we want to build a model that mimics behavior to intelligence, the output. Because the brain can compute a lot of stuff, but then how it decides what behavior to express at any given point in time, it also creates a bottleneck. Now, of course, you can think of it that if I go down to the muscles and I record the movements and everything, I basically can do everything neurally, right? I don't need to even record the phonemes that come out if I go and understand. But ultimately, you want to record functionally, the entire path. Now, if you want to drop one level below and you will say, okay, I'm going to replicate the brain at the morphological, anatomical, but physical methods, there's a lot of issues there, even technical issues, that even if we had all the data at the basically greatest detail, there will be a lot of parameters that we have to feed. And we do not have the computational, it's like we're adding complexity without having the machine learning algorithms that would fit this data. Whereas what we're lucky in a way is because we have backpropagation and essentially these artificial neural networks, and we have the way to basically feed the data to the model at this level. And because these neural networks are universal functional approximators, we are in a unique opportunity to basically copy the brain, both at the activity level and the behavioral level, without having to worry about all the detail about physical implementation. Of course, in the future, these are going to become more and more important. As I said, if you're going to fix a disease and you're going to attack the ion channels, you have to build models that link these things together. It doesn't necessarily have to be bottom-up models, but more like this type of foundation models I discussed.
I think that is all the time we have for questions today. So thank you so much, Andreas, for coming on the series. Thank you. And thank you everyone else for your time and your attention. And we will see you next month.